All right, how's it going, guys? Hope you had a great weekend and welcome back. I want to go through with you real quickly here how to add the carbon dioxide to your jars in the back and how to get those all sealed up so that we can begin to watch these plants grow with some elevated carbon dioxide levels. So, to begin with, you are going to go to the front of the room and Mr. Hansen will give you a couple of things. One, he's going to give you an Alka-Seltzer tablet. Alka-Seltzer. And two, he's going to let you tear off a sheet of saran wrap. And he's going to give you a rubber band. Sorry about the handwriting there. What you're going to do is one of you will get ready to drop this Alka-Seltzer into your cup of water inside the jar. But before you do that, make sure someone else is ready with the saran wrap so that as soon as you drop that into the water, it's going to start fizzing and bubbling and letting carbon dioxide go. Someone needs to take that saran wrap and place it over the top of the lid, and then somebody else or the same person grab that rubber band and snap that around the saran wrap to hold it on there airtight. So really the only big thing here is trying to get that jar sealed up as quickly as you can after you drop in the carbon dioxide tablet or the Alka-Seltzer tablet. Once that's done, you're pretty much done. Get the jars put back on the light rack. I know that everything is really crowded back there. Hopefully you can kind of see what I did to get all of those jars to fit back there. Do me a favor, any of the jars that are glass, make sure that they are sitting all the way down in the trays, and just if you have to set some jars up on the edge like they are right now, just do that with the plastic ones just in case somebody accidentally knocks one off of there. But take a look at where yours are at when you go back there and do your best to put them back in the same place when you're done. After that, you need to deal with your materials and methods. On your materials and methods, of course, you should have your list of materials already. But on your methods, you'll want to add what you did today to what you already have. And again, make sure you're sharing these methods with everybody so that if there happens to be a day where you are gone, if you're the writer, everyone can still work on those. Once those methods are complete, then we want to go ahead and begin addressing this week's tasks, which is what I'll talk to you about next here, section 5 of this chapter. So go ahead and pause this video, take care of your lab and your Alka-Seltzer work, and then come on back to this video and I'll get you going on this week's stuff. Alright, well, welcome back. Hopefully all that went pretty smoothly for you. Let's talk a little bit about section 5 of chapter 3 here. This section deals with cell division, how the cells divide to form new cells, and also the structure of a DNA molecule. For today, we're going to focus just on the cell cycle or cell division. There are actually a few different ways that cells can divide. We're going to focus today on one called mitosis. Mitosis is a type of cell division in which the two new cells that you get when a cell divides, they are identical to the one that you started with. So this is cell division in which the two new cells are identical to the original cell. And that's important here. That's your definition of mitosis. There are other types of cell division where the new cells you get are actually a little different from the original cell, or what we would call the parent cell. So this process takes place in stages, and I'm going to take you through each of those stages now. And I'd like for you to be paying attention to what's going on inside the cell at each of these stages. And as we go through the video here, you may want to pause and take some notes for yourself. All right, well, it occurs to me that before we can really begin to talk about the cell cycle and cell division, we need to talk a little bit about these long molecules of DNA that are in the nucleus. When you look at one of these long molecules of DNA, occasionally the cell will wrap them up, kind of like a spool of yarn, and when they're all wrapped up, they form this structure that looks something like a boomerang. And this is a chromosome. And in the middle you have this protein structure that the DNA is kind of wrapped up on. This is a chromosome. My arrow, that's not a very good arrow. Let's try that again. This is a chromosome. Normally this is what the chromosomes look like. This arm of the chromosome here is called the chromatid. That's what the arm part is. And there's a protein ball in the middle here that the DNA is kind of wound up on. That is called the centromere. When the cell gets ready to divide, one of the very first things that it does is it makes a copy of its DNA. And once a copy is made, when it winds the DNA up again, it doesn't look like this anymore. Instead, the chromosome looks like a big X. And that's because you now have two copies of this huge molecule, and they're connected together at the centromere. And this copy over here is referred to as the sister chromatid. And in fact, you'll see textbooks where they refer to the chromatids of a chromosome, and it looks like this, as sister chromatids. And once the DNA is all wound up, and again, it's good to imagine this as spools of yarn, which if you unwound them would be incredibly long. The spool of yarn analogy, I think, really helps to understand what's happened here. So this is a molecule of DNA in the cell that has been copied, and it's all wound up, or condensed, they call it. You might find textbooks that call this a condensed chromosome. It's not all unwound, it's all wound up in this nice little package, and now the cell can do what it wants with that DNA molecule, move it around to where it needs to go, and things like that. So that's the structure of a chromosome, and that is definitely test material. Hint, hint, subtle, subtle. So let's talk about now the cell cycle here. The cell cycle happens in phases, the first of which is a phase called interphase. The cell actually spends most of its life here in interphase. You've got the cell, you've got the nucleus. And within the nucleus, the chromosomes are actually all unwound. And so when you stain the nucleus, you don't really see those condensed chromosomes. Instead, this is just full of unwound chromosomes, and through a microscope like ours, it just stains the nucleus solid purple. I don't know if you remember that from the cheek cell lab, but all that DNA got stained, the DNA is unwound, and here's where the cell spends most of its life. So, what do you need to know about interphase? Most of the cell's life is spent here. There's a reason the DNA is all unwound. It's so that the cell has access to it and can be reading the information there to figure out how to do whatever it is that it needs to do. Now, one crucial thing does happen in interphase before the cell gets ready to divide. Right at the end of interphase, the DNA is copied. Oops, that is not a C. Sorry about that. The DNA is copied. And then once the DNA is copied, the process continues on from there. Phase 2 is a phase called prophase. During prophase, the membrane around the nucleus starts to dissolve. It's falling apart. So the nuclear membrane is starting to fall apart, and the chromosomes, they get all wound up into those nifty little X-looking structures that I drew for you just a little bit ago. And all living things have a different number of chromosomes. This one just doesn't look very good to me. That's all right. They all have a different number of chromosomes. This fake organism that I'm drawing for you now, we're going to say has 4 chromosomes. So, what's happening here in prophase? The nuclear membrane is dissolving, and the chromosomes are being wound up, which we call condensing. So the chromosomes condense. That's what happens in prophase. Nuclear membrane dissolves, chromosomes condense. Next, we have a phase called metaphase. During metaphase, the chromosomes line up, single file, down the middle of the cell. I always liked metaphase when I was in school. It's the easiest to remember because meta sounded like middle, and the chromosomes lined up down the middle of the cell. How do they get there? The cell actually creates these protein fibers called spindle fibers that attach to the centromeres of the chromosomes and move them wherever they need to be. So in metaphase, spindle fibers form, and they line the chromosomes up down the middle of the cell. Chromosomes line up down the middle of the cell. That is metaphase. Moving on, after metaphase, we have a phase called anaphase. During anaphase, those protein spindle fibers that had a hold of the chromosomes at the centromere, they contract. And when they contract in opposite directions, those chromosomes, they break right at the centromere. And each sister chromatid ends up being pulled to the opposite side of the cell from the other chromatid, from each chromosome. So, chromosomes break, sorry. They break at the centromere. And sister chromatids move to opposite sides of the cell. Okay, so chromosomes are broken, sister chromatids are separated from each other and moved to opposite sides of the cell. Next, we have a phase called telophase. During telophase, brand new nuclear membranes form around each set of chromatids. New membranes form around each set of chromatids. Now remember, this little boomerang shape is what a chromosome normally looks like, okay, before the DNA is copied. And remember that each sister chromatid is an identical copy of the other one. So what we have here within these two brand new nuclei are identical copies of the cell's original DNA. Okay, so keep that in mind. We have original DNA copied now. We have these two nuclei, and each one contains a copy of the cell's DNA. Finally, we have something called cytokinesis, which is not really a phase of cell division. It's more like a separate event. Cytokinesis, how do you spell that? I think it's ISIS, but it could be ESIS. I'll have to look it up later. Cytokinesis happens. You have this cell that's got these two nuclei in it. The cell divides right down the middle to give two new cells. We call these daughter cells. Each one of them has its own nucleus now, which contains the 4 original chromosomes. Those chromosomes will now unwind, and each of these daughter cells will go into interphase, where they'll hang out for quite a while. This is the actual cell division. And there you have it. These two daughter cells identical to the parent cell. I know they don't look exactly like that first drawing of the interface cell, but that's because the chromosomes are still all wrapped up, all condensed. But these are just about to unwind now as these cells each go into interphase, and they will look exactly like that first cell that we drew. So that's mitosis. All right, let's wrap things up here by talking about your chapter tasks for this week. First, I would like you to design a set of flashcards to help you study the phases of mitosis. I leave it up to you to find an application in the App Store, or do them on paper, or do them in any other app that you already have. I think there might be some cool flashcard apps out there. I'll leave it up to you to explore resources for doing this, but I will tell you what I want on each side of the flashcards. On one side, I want a drawing of the cell with the name of the phase it's in. So for example, prophase. If you're doing flashcards for prophase, I want the name prophase on one side, along with a drawing of the cell in prophase, nucleus dissolving, chromosomes condensing. I guess they'd be double-stranded at that point. Something like that. And on the other side, I do not want the identification of the phase. Instead, I want a description of what's happening. Membrane dissolving. Chromosomes condensing. You get the idea. One flashcard for each phase, front and back. On the front, name the phase, give a drawing. On the back, describe what's happening, and that way you can use these to study from either direction and do that with your group. Second task. I would like you to practice making bar graphs and data tables. And there is an activity for you to do in Schoology, which will take you through a review of how to do each of those things and give you a chance to practice each one. And finally, third, you have your favorite. Section 5, directed reading. Take a moment to explore each of these three. Over the course of Tuesday, Wednesday, and Thursday, you need to complete all three of those. They will be due Thursday at 4 o'clock. So, again, have some fun with the flashcard one. Definitely do your best on the bar graph and data tables, and just hammer through that directed reading on whichever day you think would be best for your group. So in the time that you have left today, please plan out your week, what you want to do tomorrow, Wednesday, and Thursday, and after that you can call it a day.